Okay, so uh, here are the outcomes from the count cells experiment. Um, and what I'm doing here, and I encourage all students to do this with their own plates, is I've arranged them into some kind of reasonable, organized fashion, where the replicas are here in each row is replica 1 and replica 2, and then dilution are the columns, so dilution step 5, 6, and 7. And then over here is my spot grid plate, which has nearly exactly the same experiment, but just in a much more compressed footprint. And either looking at the spot grid plate or at these plates, we notice some striking patterns. And one pattern that you should notice has to do with dilution factor. As the dilution factor increases, as the dilution step increases, we get fewer and fewer cells. And the other pattern that should be apparent is that the replicas should look similar, not identical, but similar. So on this dilution step 7, I see maybe 2 colonies, and then over here on this replica plate, I see 0 colonies. Well, when you get to small numbers, there's some probability that when you sample 200 microliters, you don't have any cells, or you have 2 cells. And if I had repeated this a few more times, I'd probably see a plate with just 1 cell. So these are the lucky 2 cells that swam into the pipette tip and got onto the plate. With these larger numbers, they start to become more, you get more confidence that you're capturing the average number of cells. But again, I'm not going to be surprised if this turns out to be a few extra cells here and a few fewer cells on this replica plate. The variation that's expected in replicas is proportional to the square root of the number of things that you're counting. So if this might be close to 100 colonies on this plate. So if this one has 90, I'm not going to freak out, because square root of 100 is 10. The average is probably 100 plus or minus 10. If over here, just eyeballing it, looks like maybe 20 cells on each plate, maybe 25 cells on each plate. So 25 average plus or minus 5. That would be just based on pure sampling, statistical probability of capturing the same number. And when you get to small numbers like this, 1 plus or minus 1 means many plates will not have any cells. And so this is one of the statistical features of the statistics of counting objects, like counting cells. The other thing I want to point out is that there's a real strong correspondence between these quantitative numbers that I'm going to capture from the counting plates and this spot grid plate. For the spot grid plate, I get the same conclusions, but everything that I can reproduce the result and that I can dilute cells, but I do it with just one plate. When you're doing your independent project, I want you to think about how you can leverage these spot grid plates, each square being equivalent to one of these larger plates, so that you can get quick preliminary findings that can guide your more quantitative studies after you know the target region. The columns might correspond to the level of stress or to the dilution factor. You want to be able to quickly gauge where in this space would be a reasonable place to now test your experiment on counting plates. So here from the spot grid plates, I see that I run out of cells at dilution step 5. And since I'm plating 2 microliters on these spot grids and 200 microliters on the counting plates, I know that if I run out of cells at step 5, I'm also going to run out of cells at step 7 for these larger volumes transferred. And then you spend fewer plates, you don't spend as much time pouring plates if you can get your preliminary data ahead of time, and then go for more quantitative results with the full-on counting plates.